Thank you very much, Christian. Ladies and gentlemen, it's nice to be here. I hear myself very loud. If I'm too loud, just do something like this. If I'm too low, do something like this. I have an option here to push a little button up and down to regulate this. So just let me know. Um, as Christian pointed out, I'm interested in host-pathogen interactions. I'm trying to understand how hosts evolve to overcome the negative effect of pathogens and how pathogens evolve to overcome the response of the hosts. And we have been doing this for a number of years already. And over the years, we have done this with different approaches. And one day I thought I would kind of give an overview over these different approaches. And I realized that the different approaches are not only fundamentally different from the way you approach the problem, it is also that you are making different assumptions in different approaches, but still you always think this is kind of feeding in the same direction. And one of the principal axes how you can line up these different approaches is by looking at the time scale on which a certain approach is relevant. And this is what you can see here. I show you this time scale. Note that it's on a log scale. We are going from today basically through different things until the age of the dinosaurs. And I will give you examples from our work which place this host-parasite interactions on different time points in this time axis. Now, before doing this, what I would like to do is to point out a few of the potential pitfalls, but also potential beautiful things which you have to encounter when you do so, because going along this time axis, as I said, you make different assumptions and you can do different types of inference. So here we have the time axis on the top, and here we have a number of different features. So for example, when you look at the left side, what you basically do is you look at the process, you look at the evolutionary dynamics, while if you go on the other side, you're looking at patterns. Here on the left side, you do experiments, you do observations. On the other side, you gather evidence by observations or by comparative analysis, comparing species or comparing genomes from different species. The temporal resolution obviously is very high here. You can go down to basically every individual generation of your antagonists, while here we have a very low temporal resolution. You may talk about microevolution on this side or macroevolution on the other side. And then an interesting aspect is the skill set or the mindset of the researcher, which you may have experienced as you talk with people, and some of them, in all their argumentation, they are kind of an evolutionary ecologist. They think about things as you would see them in the field, and they like to get their boots muddy to study the organisms. Well, on the other side, you have the people who are trained with computational biology, they do phylogenomics, they do bioinformatics, and they don't get their boots muddy anymore. And there are completely different mindsets in between. So all of this is things you need to keep in mind when you look at the experiment I'm showing you now, going over the different timescales. Now, I want to focus on one particular aspect, namely on the red-queen model. The red-queen model is an idea that hosts and parasites evolve with each other in a way that selection is frequency dependent. And an important aspect of the red-queen model is that we have an underlying matrix of this sort here, where you have an interaction between a parasite and a host. You have alleles on the locus in the host, alleles on the locus in the parasite, and the interaction of these alleles determines to what degree the interaction is compatible, so the host is susceptible and the parasite can infect, or whether the host is resistant and the parasite cannot infect. And if you have such a combination as you have it here, it's quite easy to figure out that neither of the two alleles in the host nor of the two alleles in the parasite can go to fixation. And this results in this famous red-queen dynamics looking like this here, where you have a host, and the host allele is very common, the parasite allele, which is able to infect this particular host here, in this case, uh, lowercase allele A for the host and uppercase B for the parasite, then the parasite is coming, but the parasite is overshooting because it has an advantage as long as this allele is in the majority. Once this is not the case anymore, the parasite goes down, and then you get these dynamics. It's important to know that these dynamics are such that because of negative frequency dependent selection, the likelihood of the extinction of one allele or the fixation of the alternative allele is strongly reduced. And this maintains genetic variation over long time periods, potentially forever. But what this forever means, we don't know. Maybe at the end of the talk, you know a little bit more than now. There are a few important aspects to consider. One thing is that I mentioned already, we are talking here about specific coevolution. Specific meaning you have a host population or host species and a parasite population or parasite species, and they are co-evolving. We are not talking here about a host species and 3 different parasites, or one parasite species and 3 different host species. Then you become in the range which is called diffuse coevolution, where you have a mix between density dependence and frequency dependence, and this becomes really difficult to understand. So here we are talking about the evolving host and the evolving parasite and one of each. The other thing is that it's a model where genes co-evolve, not species. So in this case here, the A locus in the host evolves with the B locus in the parasite. And this is important because what it means is that the rest of the genome can do other things. Because you have recombination, you have different chromosomes, so you have different loci. You may have directional selection for salinity tolerance or temperature in part of the genome, while at the same time, at this locus here, you have negative frequency dependent selection of host-parasite interactions. So it's the genes which co-evolve, not the species. So it's really a focus on this. I give you an example of how you may look into this different time scale things. So if you study processes, for example, here, this is actually the absence of red-green dynamics. If you have monocultures, and the notorious sensitivity of monocultures to pests and parasites kind of infecting them, this is something which you would study on this end here of the spectrum. You basically can do this in real time. But then you may compare different populations, in this case here, MHC in different flamingo populations, and you can study from the distribution of the genetic variation, you can make inference. This is still the same species, but then you may also go beyond the species barrier to a case like this here, where we have here the primates, with humans here, chimp, gorilla, and so on. And here you have a gene tree of a gene which is important in the interaction with pathogens. It's part of the MHC. And you can see from this gene tree here that there's so-called trans-species polymorphism. That polymorphisms you see in these genes are shared in different host species. You see there are completely different ways of how we look at it, but three times we still talk and think about red-green coevolution. That's what I mean by you're using different assumptions. What's also important is, while here on this end, on the left side, on the now, we know who is the parasite, the further we go into the past, we typically know only one of the 2 antagonists, either the host or the parasite. So when we talk about here, for example, trans-species polymorphism, we don't know who was the parasite selecting there. We can make speculations about this, but the inference you do here, for this you don't know who this was. So it's actually of interest to ask the question, who was the antagonist? And the other thing I would like to ask is whether the process of now is the same as across time in the past. Now, the work we do mostly with this particular model species here. This is Daphnia. Daphnia magna is a species. It's a water flea. It's a lower crustacean. So it's actually closer related to Drosophila than it's to lobster, even though they are both crustaceans. This is an uninfected female here. You see the female has this eggs in the brood pouch. The eggs are asexual eggs. They can produce monozygotic twins in any number, and the offspring are exactly identical to the mother. But you can also cross them. They also reproduce sexually. Roughly, you have one sexual generation per year, but multiple asexual generations per year. On the other side here, you have an animal which is infected with a parasite. The parasite is called Pasteuria ramosa. And this is how the spores of the parasite look like. They're quite big. They're about 5 micrometer in size. You can just use a normal light microscope to see them. It's very easy to work with them, even though they are bacteria. And this Pasteuria, they infect the host, and then they proliferate inside the host, and they castrate the host. This is why you don't see any eggs in the infected female, and the host then starts to grow. It becomes bigger than the uninfected host, and it eventually becomes just a sac packed full, filled with transmission stages of the parasite. So it's highly virulent because the host does not reproduce anymore. An infected host has only about 10% of its residual lifetime fecundity left. So it's really very detrimental for the host. And it's widespread. On the lower part here, you can see this light of the Earth. In green is the area where Daphnia magna occurs, here, also here in Ethiopia, in South Africa, and in also South Africa, Siberia, Europe, and North America, we found the parasite. We find it quite widespread. Not every population has it, but many populations have it. So it's really a common parasite throughout the species range of Daphnia magna. So let me start first by talking about one of the very important assumptions of the red queen model, namely the genetic variation which you should have for it, the so-called standing genetic variation. This was a very important publication we published already 25 years ago. Excuse me that I'm showing you such old things here, but I still find this quite inspiring. We went to one population and we collected 9 Daphnia magna. They were all infected with the parasite. We isolated the parasite, we cured the host and multiplied the host clonally. So we had no clonal replicates of the host and clonal replicates of the parasite. And then we made all possible combinations and tested their interaction. And each cell in this matrix here is a percentage calculated from 18 replicates per cell. So this means 0 of 18 replicates became infected. This means all 100% of 18 replicates became infected. What you can see there is that there's enormous amount of variation. This is not the type of variation you would expect if there is a selective sweep type of scenario where a resistant parasite spreads through the population or a susceptible or an infectious parasite is sweeping through the population. This is something different. We have here much, much more genetic variation than you would expect from other loci. And actually, we looked at other loci and they are much, much less variable. So this was our first direct evidence that there must be something of a red queen type model explaining what we see here. I'm jumping now many years forward. This is a method which had been developed actually in human genetics to understand the specific interaction between hosts and parasites. What the first people did is they went to a hospital and they took samples from humans which were infected with HIV, and then they sequenced the patient plus the HIV together. And when you do so, you can then later on produce a matrix like this here, where you line up all the SNPs of the parasite genome and all the SNPs of the host genome. And then you basically make for each combination of the SNPs, you make a logistic regression and see whether there's an association between the two. And an association would be something of this sort here. It doesn't need to look exactly like this, but it would look somehow approximately like this. The important thing is you don't get a significant hit when there's a main effect. So it's only when there's an interaction effect that you get a hit here. So you would find places which have a significant hit, and it means the SNP here in the parasite and the SNP here in the host, they are associated in this form with each other. So we didn't do this with plants, we did this with Daphnia. So this was the experimental design, 250 Daphnia from one population collected, and then we sequenced the Daphnia and the parasite together. And this is now the way I present you the data. Let me walk you through what this graph actually means here. I guess most of you know what a Manhattan plot is. In a Manhattan plot, you have a genome lined up on the x-axis, and on the y-axis, you have some kind of a statistic which tells you about the strength of an association. Now think about a Manhattan plot as being a graph where you have all these peaks, and now we cut off the upper part. So we actually see only the tops of the skyscrapers. This is what you see here. So this is kind of the upper part of the Manhattan plot for the host, and this is the upper part of the Manhattan plot for the parasite. The outer circle with the bluish background is nucleotide diversity. If it's higher, like here, for example, this means there's high nucleotide diversity, or here there's low, this is low nucleotide diversity. And so this was our first result, what we get. Here in the locus, at this place here, and you see there's high genetic diversity, we have a highly significant hit in association with this cluster of genes here in the parasite, where there's also high genetic diversity. What is interesting is, you see there's a C written here. It turns out that this is a C locus, and we knew the C locus before, segregating in this population. So it was nice that we find something we know already. This confirmed that the method actually is able to discover something which we had evidence before. More important, we didn't know the parasite side. So we got the host side, confirmed what we had, the parasite side was new. Second about the C locus is that the C locus has very strong interaction, epistatic interaction with other loci. So basically it makes the effect of other loci invisible. But since we knew this, we could now correct for it in the analysis, and this we did. And this is now taking the C locus out, and you see now the interactions we got. You see there are many more interactions. You see again 2 letters here, D and E, and this again, these are 2 letters we knew before. So again, it shows that we uncover what we had known before, but again, we didn't know the corresponding site on the parasite. So this was nice. Here we discovered a new locus, which we had not seen before, and the interacting site on the parasite. What you can also see here is that wherever on the parasite this line ends, there is a peak in diversity here, 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 here. So the parasite has this diversity, highest nucleotide diversity at the places where the interaction is with the host. What you also can see here is that we marked them in red, and this is one particular gene family. These are collagen-like genes. These genes are responsible for the attachment of the parasite to the host. So we basically mapped with the system here also the genes in the parasite which are responsible for infectivity. The final thing I want to show you is that this here looks a little bit messy. You have the impression that there are many loci sitting beside each other. As a matter of fact, this is one locus, but the locus is 5 MB in size. It's a haplotype block which is not recombining. When we do individual mapping experiments, it maps sometimes here, sometimes here, sometimes here in within this 5 MB, just depending on the idiosyncrasies of the frequencies of some SNPs in this block. So actually all of this here, we call it the E locus, is one locus segregating in a Mendelian fashion, but it's a huge region. We don't know what the gene in this region is responsible for the resistance, but it's just one thing. So in the end, what we have is we have the C locus, the D locus, the E locus being one, and this one here. So 4 genes on the side of the host, and on the parasite side, we have 1, 2, 3, 4, 5 the correspondence. For this particular population, these are the genes which are co-evolving. If you go into the genetics and do breeding experiments, you can uncover that indeed this matrix, as I showed you before, is indeed verified in this breeding experiment with this loci. This here is with the host variability and 2 parasite isolates. This here is with different alleles in the parasite, and you can see that they are getting, this is the stuff which is producing the interaction. So to sum up this part, understanding genetic variation, we have very strong allele-specific interactions and large standing variation for host-parasite interaction. So this is the stuff you need that red-queen dynamics can work. Without this, no dynamics. So now let's move back in our timescales. We start with from months to years. I show you here an example from a pond which we investigate in Switzerland since a couple of years. You have here one season in 2014. This year is the prevalence of the parasite. You see in this year it went to nearly 100% of the adult animals, and was only there in summer, in April, until mid-March it was not there. Here you have the resistor-type profile of the host. In blue, these are 2 different resistant hosts, and in yellow, 2 different susceptible hosts. And you can see in the beginning, we have about 50% susceptible hosts. Then the parasite kicks in, and then susceptibility goes down. And by the end of the season, all the susceptible ones are gone, which indicates how strong selection is in the system. I mentioned before the parasite castrates the host, and this is the signature of this. The infected hosts don't reproduce anymore, and you are left with only the resistant ones. Now the surprise was this. We were here at 0, then the winter is coming. In winter, the Daphnia are not there, they're in resting eggs, and then we are back to 50%. Then again, the parasite kicks in, we go down to nearly 0%. Again, winter, again, next season, we are at 50%. The parasite kicks in, it goes down, and so on, year after year. This is a phenomenon we are still not able to explain. We can explain part of it with the epistatic interactions between hosts and parasites, but only part of it, not the whole spectrum. So this makes it a little bit tricky for us to understand what's happening there. It looks as if we are seeing cycles and we are reminded to red-queen cycles, but these are not red-queen cycles. These are cycles driven by something else, partially by what is called the slippage of sex, which is basically caused by alleles are hiding in genotypes and being protected from selection, like when you have dominant or recessiveness, and epistasis. So this explains about 50% of the variation we see here, but there's still 50% we need to explain and we can't. So there's still something to go on. But the important point is we do see strong selection over time scales, which make sense for how the red-queen dynamics were suggested to work. We did another study which goes further back in time. You see, we are now here several decades in time by taking advantage of the fact that Daphnia is producing these resting stages here. They're the sexual eggs of the Daphnia, and they can survive long periods in sediments. And the parasite as well, this transmission stages of the parasite, they can survive decades in the sediments. So we have both the host and the parasite surviving in sediments. So we can take a sediment core, which is basically you take a plastic tube, stick it into the sediment, close it, and pull it out. You have the sediment core, you slice it, and the slices are time periods. And then you bring the Daphnia back to life and the parasite back to life. Then you do experiments as if you had collected the Daphnia 30 years ago. And we did this, we did a so-called time shift experiment where we take the Daphnia from one layer and infected them with its own parasite, the contemporary one, with the future ones, and with the past. And so there's a certain expectation. If this is red queen dynamics, the dynamics of these three time layers should produce a shape like this, where the highest infectivity is in the contemporary ones, lower one in the past, lower one in the future. And that's exactly what we get here. So this is for the time layers of two different sediment cores, which are combined here. And you see, indeed, on average, we get this pattern here. There's an alternative model, I don't want to talk in detail about this, which would produce something like this. So this would be a significant different thing, and this would be a different way how coevolution goes. But since you get this, this is supportive of red queen dynamics. Now let's move a bit further into the part. Let's move towards the ice ages. Towards the ice ages, all of Europe was covered with ice, and there was no space for Daphnia here, no habitat for Daphnia, I should say. The Daphnia were hiding out somewhere here in southeastern Europe, in the Middle East, not being able to conquer Europe. But then about 10,000 years ago, the ice disappeared quite rapidly, actually, and then Daphnia moved all over Europe and were spread out now all over Europe. Now, this means we can now use a space for time replacement, assuming that in different populations, coevolution happened kind of in a similar way, so that what you see in different places resembles each other in a wider frame. So we did this, and the first thing what we did here is we collected Daphnia from all over Europe, or the Western Palearctic, I should say, and then we tested them against this parasite C1 here. And red means the host was susceptible, blue means the host was resistant. And if there is balancing selection, you expect that there is a uniform distribution of red and blue. And this is what the red queen predicts, and this is what we do find indeed. It's really a quite nice uniform distribution of resistance and susceptibility. The other way around is to do this with a parasite. So now we took one host clone and infected it with different isolates from the parasite collected in the Western Palearctic. And you see again red and blue, and it's again more or less evenly distributed red and blue, uniform distribution. To give you an idea how you should think about this, imagine you would have taken it, you would flying over Europe and you would collect from every city one individual human being and just ask, does this human being have a Y chromosome or not? And then if it has a Y chromosome, you make a blue triangle. If it has a no Y chromosome, you make a red triangle. You would get a uniform distribution because in every village, in every city, you find approximately even numbers of males and females, carriers of Y chromosomes or carriers of X chromosomes only. Now there's an alternative to this here. It could be that when the ice disappeared from Europe, that this was so quick that the Daphnia just flooded all over Europe in one blink of the eye, so to say, and that all of Europe was basically just the signature of this splash of Daphnia all over Europe. If this would be the case, all other genetic markers should show the same signature here. But that's not the case. What we find is that we have a pattern of what is called isolation by distance. So geographic distance is shown here, and this is pairwise relatedness of the Daphnia genotypes, which we had sequenced from the different populations. And you see we get a nice decline of relatedness over distance. This is a signature of isolation by distance. Here we don't see this. There is no isolation by distance. Here we see this. A different way of looking at it is to look at other traits. Remember I told you that the red-queen dynamic is about coevolution at specific loci. Other loci may evolve differently, and they may evolve in response to the climatic conditions in Europe. So we check this, and you can see here different features. For example, this is the length of the spina. Daphnia have a little tail spine here. You can see this there, here. And you see when you go, this is across the entire Western Palearctic plus Asia, and you see Daphnia in the north have a short spine and the south have a long spine. So there's local adaptation for predator defense, which you can see across this area. Or likewise here, this is body size. Daphnia in the west are small, in the east are bigger, and there's a gradient in this direction. You can see already these two gradients don't agree with each other. Very likely different genes playing a role to determine this. And so you evolve local adaptation in one direction or in the other. Or yet here another example. This is the color of the resting stages. They can be black or more whitish. And you see there's yet another shape of the distribution. So you see that different features evolve differently from each other, indicating that there is no reason to assume that this here is caused by the initial wave of Daphnia across Europe after the Ice Age. So now we can move even further back. Daphnia shows three subclades. You may call them subspecies or so. There's no agreement on what they actually are. You have a North American one here, you have an East Asian one, and you have a European one. And we collected Daphnia samples from all of those. This is part of our Daphnia diversity panel. And if you look at neutral markers, you get exactly such a tree here. So it's very clearly that the background, the backbone of the Daphnia genome shows you this clear separation, these three different clades. If you look for resistance patterns, so it's the same now as before, you see that we have resistance and susceptibility all over the entire spectrum. Even here in South Africa, we have only two populations which we discovered there. We have both different resistant and susceptible. And you see here everywhere across the entire space. If you now look how these different subspecies are distributed here, you can see that there's not much of a difference between here and here and here. You would not call this a different subspecies based on the distribution of resistance or susceptibility. The polymorphism causing this resistance polymorphism is spread across this different subspecies. Now you might say, okay, well, this is one parasite isolate, it's called P50, but there might be it might look other for different isolates. So we did this with 14 different isolates. These are in total more than 30,000 tests we did, or I should say Michael Krebs did this. And what you can see here, in every single case, we find that we get the same uniform distribution. There are cases where you have more red, there are cases where you have more blue, but it's always the polymorphism is always present in all three subspecies throughout every single case. We had not a single case where the polymorphism was absent from one of the three subspecies. So again, strong evidence for balancing selection on resistance genes, consistent with the prediction of the red-green model. Now let's even go further back to the past. We are now crossing the species barrier. We are now going to species which are related to Daphnia magna. What we are looking for is a pattern called transspecies polymorphism. Transspecies polymorphism occurs when you have a scenario like here. We have an ancestral species which splits into two new species, so host 1, host 2. And in the ancestor, we have an new allele arising through mutations, and then you have the capital H and the lowercase a allele. And this polymorphism is maintained and is maintained in each subspecies. As a consequence, the red allele here and the red allele here are closer related to each other than the red allele and the blue allele within the same species. If you would do this with other loci, which are not under balancing selection, you would always find that the gene tree matches the species tree. If you have transspecies polymorphism, the gene tree, shown here, does not match the species tree. You see here host 1, host 2, they're both in one clade, and here host 2, host 1 in the same clade. So we tested this with these three species here, Daphnia magna and then Daphnia sinensis similis. They're not the direct sister species of Daphnia magna. There's one species in between, but it's only known from one population, so we didn't use this. But all these three species here are very widely spread. So we collected many individuals, sequenced all the genomes, and then tested for this. Here is the species tree, together with the timing, but this is the minimum age of the split. So it's at least 15 million years ago that this clade split from this clade. And then here you have 2 of the 4 regions we discovered which have a shared ancient polymorphism. So you see in each clade here, you see all 3 colors here, up here, and the same here and here. We did this in a candidate-free way. So we screened the entire genome and did some very strong filtering, and in the end came up with 4 regions which shows transspecies polymorphism. These 2 regions here, one of them sits inside a posterior resistant locus and the other just outside. And from the other 2, one is sitting very close to another posterior resistant locus, and the 4th one sits somewhere else. We don't know what it is doing. It might have a completely different function. But this means that 3 of the loci showing transspecies polymorphism are actually associated with resistance to the parasite we are talking about here, suggesting that it is indeed Pasteurella which is causing this long-term transspecies polymorphism. So if I sum up here the evidence I showed you on the timeline, so we have here evidence for balancing selection of resistant genes in the Daphnia going back to 15 million years. And I can just announce we have now evidence that the loci are possibly 200 million years old. So we would go much, much further back. We recently discovered in related species that the same loci are sitting there segregating. We cannot say yet whether they have also to do with resistance to Pasteurella, but at least the loci are there and they show the same genomic signature. The parasite, Pasteurella, we have good evidence for about 1.5 million years ago, indirect evidence until 15 million years ago. And there we also have just new comparative evidence with related parasites that this is also possibly even going further back. So very clearly we are talking about a system which is co-evolving for a long time, and it's specific one-to-one co-evolution which is happening here in this particular system, meaning that the same antagonists can co-evolve along long time periods. And the process which we looked at here is well able to explain the deep time patterns. Now, in the beginning, I said that the red-green dynamics, one of the important things is that it maintains genetic variation. And this is not just one of this kind of nerdy prediction. This is possibly the very, very most important feature of the red-green dynamics. And if you look into the literature and you Google coevolution, what you find is you find an enormous amount of people talking about red-green dynamics. Even so, the evidence for red-green dynamics is comparatively thin. So what I showed you here is possibly among the best evidence existing worldwide. So why do so many people talk about red-queen dynamics if the evidence is actually not as good as you would hope for a theory which is 50 years old? The reason is the red-queen dynamic is the only mechanism which can explain a feature which we see everywhere and we cannot explain otherwise, namely extreme genetic diversity at regions in the genome which are associated with parasitic disease. Think about the MHC in jawed vertebrates, think about other genes in plants and other organisms. So how does this look like here? I showed you already a little bit of this, but I want to stress this a bit more. This here is the sliding window approach over the genome of Daphnia magna, over many genomes of Daphnia magna. And then we calculated the nucleotide diversity for each window, and you can see a histogram. You see the median for the genome is somewhere at about 8 SNPs per 1,000. For comparison, humans are at about 1 SNP per 1,000. So Daphnia is much more diverse than humans. But I saw today data from Drosophila, and it looked at Drosophila is actually a bit more. It was kind of, I think, 11 SNPs per 1,000, which I found quite impressive. Our resistant genes are sitting here. So they're far outside on the extreme side. We can do the same for the parasite. The parasite is remarkably invariable. So we have here much, much lower rates of nucleotide diversity, but the resistant genes or the infectivity genes are far out here. So indeed, the resistant genes and the host and the infectivity gene and the parasite are on the extreme side of the diversity spectrum. Now, there's an interesting aspect here, namely, what I show you here is actually cheating. Cheating for the host, not for the parasite. For the parasite, this is what you get. For the host, the regions where the resistant genes are sitting in are so diverse, we cannot align them. When you cannot align them, you cannot calculate nucleotide diversity. So what we are doing is we look at the flanking regions. These are the flanking regions. So the flanking regions are already super diverse. What about the regions themselves? This is one of them. This is a so-called dot plot. In a dot plot, you take part of a genome, you line it up on one axis, you take the equivalent part of the genome from a different genotype, put it on the other axis, and if they do what genomes normally do, they show homologous sequences, then you get a diagonal line. What you can see here, nice diagonal line, and then suddenly you get this hole here with tiny little lines here which are repeats, but clearly no diagonal. And then it continues here with a diagonal line in this upper corner here. The region in between there where the hole is so diverse that there's basically no homology between haplotypes. And initially we thought, okay, maybe there are 2 haplotypes in the population. We sequenced 14 more genomes, and we have to do this with long read, high fidelity sequencing to get the proper readout there because of all the repeats. What did we get? 14 more haplotypes. This region is super diverse. That's one way you can look at it. A different way to look at it is this one here, at another region. This is actually one of the regions which showed up in this transpecies polymorphism. We have now here again the positions lined up on the chromosome, but this is a so-called LD plot. An LD plot looks for linkage disequilibrium. And LD normally for regions which are far away on a genome would be close to 0. And this is what this gray area here is showing. If you are close to each other, you get red, or a little bit further away, yellow color here. And then suddenly you get this huge square here. This is a non-recombining linkage block of about 5. Well, depending on, so you see it's the size here is about 14 MB in size. In the middle is a block of about 5 MB, which is like this one here, completely non-homologous. And the reason it's white is because we cannot do calculations of LD anymore, because we cannot align them. Something weird is happening here. We don't know what it is. We also sequenced here multiple genotypes, and again, every genotype is sequenced from a different population had a different haplotype. For one particular population, we sequenced many, and there we found there are 3 haplotypes within this population, and they are segregating with each other. And actually, it turns out that this is what is causing this dynamics here. So this is the changes in the frequency of the 3 haplotypes underlying this non-recombining haplotype block here. So there's something major going on in this region there, affecting a large part of the chromosome. They're sitting 400 genes in this region. So we're not talking about peanut. There's quite a bit going on here. So we have a scenario where we have negative frequency dependent selection. We have balancing selection. One might say, well, this means that actually not much is happening, much ado about nothing, because you basically cycle your frequencies all the time. Is it really like this? The answer is yes, with regard to the functional genes which are causing this polymorphism, but the answer is no for what's sitting around it, because these things are sitting in regions which have a certain recombination rate. There might be recombination blocking things around this, so they influence an entire part of the genome. So for the host, we have massive structural polymorphism around the resistance haplotypes. About 8% of the whole gene is affected, and 2% of all genes are sitting in affected regions. Given that I showed you before, 4 genes in the host are responsible for the interactions, 2% of all genes in the genome are affected by these 4 genes. So we're really talking about a major structural thing here happening. For the side of the parasite, we have this massive expansion of the collagen-like gene family with about 50 members, and they play a role for the infectivity. About 5% of the parasite genome is affected by this. And then we have very strong evolutionary dynamics. And interesting, we found now out that this collagen-like gene family is mainly evolving by transposition. And transposition, of course, affects not only these genes, but also the rest of the genome. So again, a major impact on the entire genome. So red-queen dynamics is more than just cycling of some SNPs. Conclusion, long-term red-queen coevolution can be a major contributor to genome evolution. I want to thank the people who contributed to this. In this slide here, the size of the font is proportional to the number of slides the students or collaborators contributed to. The same also here for the collaborators from outside Basel University and the people funding my research. And I thank you for your attention.